Hello, everyone. My name is Dr. Paul Richardson, and I'm the Director of Clinical Research and the Clinical Program Leader here at the Jerome Multiple Myeloma Center at Dana-Farber Cancer Institute in Boston, Massachusetts. And I also serve as the R.J. Corman Professor of Medicine at Harvard Medical School. And it's my pleasure to today discuss with you what I think are very exciting implications from the MIDAS study led by our French colleagues, and in particular, Dr. Aurore Perreault as the principal investigator, with her senior investigator, Dr. Philippe Moreau. And in this study, our French colleagues sought to explore the impact of isatuximab, carfilzomib, lenalidomide, dexamethasone as an induction strategy in newly diagnosed myeloma for patients who are transplant eligible, and explore that impact on MRD negativity. And then in a very, I think, forward-thinking and innovative fashion, understand the impact of high-dose melphalan on a surrogate for clinical outcome, namely MRD rate, in terms of potential therapeutic benefit. So the first clue we had to this data was published in Blood earlier this year by Aurore and her colleagues, where they were able to show that in patients with newly diagnosed myeloma, the use of, and these were transplant eligible, basically almost 760 patients treated with the induction therapy. And what we were able to show was, number one, that, or what they were able to show, I should say, is number one, that they were able to get an excellent CD34 stem cell yield, and also to achieve remarkably high rates of response. So the best overall response rate was 95%. The VGPR rate or better was a striking 91%. And during induction, unfortunately, they did have 7 patients who did progress. They did have 5 deaths, mainly in the setting of progression and unfortunately cardiac events, which probably related to carfilzomib, but fortunately a very small number overall. What was really interesting to me was that MRD negativity was particularly high. 63% at 10 to the minus 5 and 47% at 10 to the minus 6, with some variation seen across ISS stage and cytogenetics, but importantly, a manageable safety profile, as alluded to, with apart from those cardiac toxicities with carfilzomib in a very small number of patients, no other major new issues identified or any new signals for that matter. Now, I think what's then really exciting was the more recent presentation by Dr. Aurore Perreault and her team at ASCO this year in June, and very importantly, their publication simultaneously in the New England Journal of Medicine, which was published as hard copy at the end of July. And it was a privilege to write an editorial together with my colleague, Dr. Nikhil Munshi, and Dr. Dan Longo, who serves as an associate editor for the New England Journal, on the findings led by Dr. Perreault, Dr. Moreau, and their colleagues. I think what's really important to note is this trial was designed to assess the impact of high-dose melphalan on quadruplet therapy outcomes. And so essentially you had a randomization between 4 arms. Basically, if you were post-induction MRD negative, you were assigned to arm A or B, which consisted either of 6 additional cycles of Isa-KRd or high-dose melphalan, 200 milligrams per meter squared with autologous stem cell reinfusion, followed by 2 cycles of Isa-KRd and then lenalidomide maintenance thereafter for up to 3 years. Now, the group who were MRD positive were considered higher risk, irrespective of their stratification, and they were MRD positive times 10 to the minus 5. In that context, these patients, and they were in fact assigned to either arm C or arm D, either received a single transplant with 2 cycles of Isa-KRd or tandem transplantation with Isa-IBA maintenance. So what's important to note is that the first group were MRD negative, had to be to enter it. That arm A consisted of 243 patients, arm B consisted of 242, so very, as expected, very equal randomization. Intriguingly, prior to maintenance, the MRD negative rate for arm A, who received 12 cycles of Isa-KRd, was 84%. And for those patients who received 8 cycles of Isa-KRd plus an autologous stem cell transplant, it was not significantly different at 86%, with an an odds ratio of 1.17 and a p-value of 0.64. So no clinical difference or meaningful difference or significant statistical difference between arm A and arm B, with the addition of a single intensification of high-dose melphalan. So this was surprising and actually counter to what was expected. Then what was also very interesting is for those patients who were MRD positive, what we saw with a single transplant plus 2 cycles of Isa-KRd versus tandem transplant, no further Isa-KRd, and then straight into Isa-IBA as maintenance, which was distinctly different. Before going into that phase, we saw that single transplant achieved an MRD rate of 40%, and that's an MRD negative rate, 10 to the minus 6 of 40%. And Arm D achieved an MRD negative rate of 30%, or 32, I beg your pardon. This was really interesting because what it showed was actually, and this was not statistically significant different, the odds ratio of 0.73 and the p-value was 0.31. But what was really interesting was that the tandem transplant arm did not outperform the single transplant arm followed by Isa-KRd. In fact, the MRD negative rate for the latter was 40%, as I mentioned, 32% for the tandem transplant. Now, this was very surprising. We had expected at least that the tandem transplant would move the MRD needle, and it didn't appear to do so. And in fact, what was important to note was that there were no new safety signals, and obviously sustained MRD negativity and PFS estimates are obviously ongoing, but the initial PFS estimates for all of the arms mirrored what we saw with the MRD, with no major differences identified between any of the arms to date. So we need to know that, as we pointed out in the editorial that accompanied this, I think, very important and pivotal paper. But we also pointed out the fact that the MRD negative rates did not meaningfully change with high-dose melphalan, either for tandem or for single, depending on MRD status, really opens the door, in my opinion, to a tailored approach to therapy. And what I mean by that is that one size doesn't fit all, and there may be important subsets of patients, as we first showed in determination, who may benefit from a transplant-sparing approach, in other words, keeping transplant in reserve, recognizing that transplant carries with it significant risks, be it acute toxicities, infections, quality of life considerations, but also, I think, most importantly, the late cancer risk, which we're realizing is more and more of a challenge as patients live longer with their myeloma, which is a great thing. But obviously the challenge of secondary MDS or AML, whilst very rare, fortunately, or rare, isn't plateauing and appears to be increasing slowly over time. So this is not trivial. What's also very important is that after high-dose melphalan, there's evidence from other studies that mutational burden at relapse from the myeloma itself may be grossly magnified. And if that's the case, this may explain why, for example, we've not seen overall survival differences between early or delayed or deferred transplant. All that being in mind, the MIDAS trial, I think, really informs us that with some degree of confidence, we can say to patients, if they're standard risk MRD negative, they can reasonably keep transplant in reserve. If they are standard risk MRD positive or high risk, regardless of MRD status, we need to think about their approach. But certainly based upon some of the quad data here, MRD negativity appears to be the game changer. And in that context, the use of it as a tailored approach to treating patients adequately, but not too much, becomes very important. We did see some very interesting subgroup analyses. Translocation 11-14 patients seemed to take more time to become MRD negative, and the MRD positive group in that particular cytogenetic subgroup didn't seem to do particularly badly. So that all didn't seem to fare worse, I should say, than their MRD negative counterparts. So this perhaps is of interest. The second important point was that translocation 4-14 positive patients did seem to derive benefit from transplant. And this is important because this is something we also saw in the setting of determination, so mirrored a previous finding in a large phase 3 trial. So putting this all together, I think we have a tailored approach to transplant supported by MIDAS and previous studies, including determination and IFM, DFCI 2009, as well as others. I think we also have the real impact of quadruplet therapy. This has been transformative. The use of a PI, an IMiD, steroids, and a monoclonal antibody targeting CD38 has truly transformed the treatment landscape for newly diagnosed myeloma. So very good news for patients, in my view, and at the same time opening the door to other options, be it immune therapies in particular, such as CAR-T, or I think very excitingly, the use of bispecifics together with next generation oral agents such as the CELMoDs and in particular iberdomide. So a very exciting study, in my opinion. I think the impact of it is truly global. and I congratulate our French colleagues, and in particular the leadership of Dr. Perot, Dr. Aurore Perot, and all of her team for an outstanding contribution to the field. Thank you.